It's live? Okay. Okay, I think we should get started without further delay. We got delayed because of some technical issues since we didn't have an in-person seminar for 2 years or so over here. And today, this is our Safari Live seminar. Hopefully many people are watching it online. And we're lucky to have first in-person seminar or hybrid seminar in 2 years. And we have Serghei Mangul, Dr. Serghei Mangul from University of Southern California. He's an assistant professor in the Department of Clinical Pharmacy.
That's correct.
Right, at the University of Southern California. And his lab is working on a lot of interesting things in biomedical research. And I'll read from here. They design, develop, and apply novel and robust data-driven computational approaches, which he will talk about, that will accelerate the diffusion of genomics and biomedical data into translational research and education. And I agree with some of the approaches, his approach in his lab. His lab believes in data analysis, transparency, effective sharing, reproducibility, software usability, and archival stability to foster a sustainable data science ecosystem in biomedical research. I actually think this is applicable to many sciences in addition to biomedical research, which is great. Serghei holds a PhD in bioinformatics from Georgia State University, and he did his bachelor's in applied mathematics from Moldova State University. And he also did a postdoctoral training with Elazar Eskin at UCLA. And he recently won the NSF Career Award. And he's going to talk about opportunities and challenges of computational data-driven immunology. And I should mention that Serghei has been a great collaborator. We've been collaborating with him for over the course of several years, and we're continuing that collaboration.
Yep.
Thank you, Serghei.
Yep.
Please go ahead.
Yep. Okay, great. Yeah, thank you, Honor. So I'm really excited to be here, and actually, especially excited. I think it was 2 years. So that's my first in-person seminar in 2 years. So that's feels surreal, but that's really exciting. Okay, so exactly. So I would like to talk about kind of computational driven immunology and kind of try to define that field and kind of talk more about that and think about what are the opportunities there and also mention the challenges. So I would like to start with the data and typically, in my opinion, data is taking the central stage across very many domains. And if you think about the domains like computational immunology, there are multiple components involved. And there is obviously the component of domain knowledge. So you need to know immunology and be aware of the knowledge there, as well as computer science and statistics, mathematics. So if you think about computational biology, which is kind of in the middle, so similar will be true for computational immunology. And I think it's the whole rise of computational immunology, the same as computational biology, is driven by enormous amount of the data being generated for the last several decades. So we have, on one hand, on the left, various technologies like genomics, epigenomics, transcriptomics, and you name it. And what they produce enormous amount of the data, which need to be handled, as well as the data is not necessarily perfect, right? You have got sequencing errors and other limitation of the technology, which needs to be overcome before kind of you can get the interpretation to the clinicians, right? So what is the in between? And in between, I really think this is the bioinformatics, or the way I see that. The noisy technology and large data sets on the left, and meaningful and easy to understand the interpretation in the translational science on the right. So why that happened? I think that's primarily due to accessibility of the technology. So that's the plot probably used so many times. But I think that's the best illustration why we're here and why we talk about omics technologies overall, just because they are scalable, right? So they are at the level of price, which is affordable for many labs in the world. So you can generate the data to answer the questions you would like to answer. And then usually we compare to the Moore law, right? So on the exponential scale, the Moore law is linear. However, I guess drop of the sequencing data is super exponential in that sense. So we are here at this stage, and that's what that result in. So that result in many labs across the world generating the data. And actually, as they do, the good convention happening right now that the data needs to be shared, right? So if you publish in a journal, most probably will be required to share our data, which is actually a great model, because this way we have accumulated quite tremendous amount of the data being available on the public repositories across a variety of diseases, across a variety of ancestry groups, and so on. We are not necessarily doing the perfect job sharing the data, especially in terms of annotations and metadata, which I will talk a little bit about. But the data is there and can be leveraged to the extent to answer interesting questions. So this is the public data we have right now. So we have two main repositories, Geo, Gene Expression Omnibus. Older one traditionally started with getting the data from gene expression arrays. And now it also accepts the data from RNA sequencing, like gene expression the data. And then we have SRA, sequencing read archive. So basically any sequencing data goes there, right? So this is the number of samples. So that's quite many, I would argue. That's a public data available as we speak, right? It's publicly available. Anybody can use that and use it to the extent to answer some interesting question across variety of domains. So, so then, given the public data being available, I think that gives us a rise to the new type of research, at least in biomedical settings, right? So traditionally, the research in biomedical settings were done in the vet lab, right? Like you got a question, you got the hypothesis, what you do, you recruit the patients or you grow the mice, you probe them with certain technology and you answer that question, right? That was a traditional vet lab research. So I think given the availability of the public data and powerful computational methods and computational power, we are talking about the dry lab. And the dry lab, where computational data-driven research is happening, where you're developing the tool or perhaps applying the tools across a variety of the data sets to make biological discoveries, which are as important as you would do in the vet lab. So this is what our lab is. So we are purely a dry lab. We don't have vet component. However, we hope that we are capable of bringing some interesting insights in the field of computational biology and computational immunology. So, and then how that is happening? So that's happening in one part due to secondary analysis of the data, right? So as the data being accumulated on the public repositories, now you can utilize that and reanalyze it again. And there are multiple reasons you would do that. One reason is sample size. Right, so individual labs are limited, even though the technologies are scalable, but they're not scalable yet at the level, let's say, of 10,000 samples. And so for the vet lab, doing the data generation in the vet lab, you will be kind of still limited in the sample size. So, and then if your effect sizes of the disease you're studying are low, you would need a lot of samples. And then what might happen in the vet lab, you will just not reach that necessary sample size individually in the vet lab. However, as let's say 10 vet lab tries that, so now the computational researchers like we are can take the data and do any sort of meta-analysis and be better positioned to discover some associations or any other kind of signal from the data, providing the insight in disease. And I think that's happened, for example, for schizophrenia, where people try for very, very many years to find association between the disease and the genetics, and they could not do that. And the reason is because you require enormous sample size. I think as soon as they reach 10,000 samples, then they found the association, right? But you never know. When you're studying the disease, you never know what is the sample size required. So meta-analysis in that sense is well positioned. Another thing which I will talk today about is about repurposing the technologies, right? So as we design the technology, we think about the specific question we would like to answer with them. For example, RNA sequencing, right? The intention of inventing or designing RNA sequencing was to study the transcriptome, obviously, right? And then kind of replace the gene expression array, which actually happened, right? Nowadays, I think majority of the transcriptomic studies are done by RNA sequencing rather than by gene expression array. However, when those technologies were designed, I would say there is never kind of only one question to be answered with the certain technologies, right? There are more than that. So, and what we can do, we can always repurpose the technology. So for example, we'll talk about immunogenomics. So basically, if you do RNA sequencing, you also, among the whole kind of spectrum of sequencing reads you're getting from a variety of the transcripts and isoforms. So what you're also getting, you're getting the reads from the T and B cell receptors. So in T and B cell receptor, they have their own genomes, which are produced by the combination of VDJ genes. So we'll talk more about that. But then having that, you can actually quantify the immune response, right? So now using the technology which was designed for transcriptomics, but now you say, okay, I can do that for immunogenomics. So that's another reason why you would do secondary analysis. I think there are a few more. So, but then, so that's kind of oversimplification, of course. So that's like more like wet lab research where you generate the data, you analyze the data, and you share the data, and you got your publication, right, and the results. So what is secondary analysis, what the dry lab will do, right? So you skip the whole part of generation of the data, so you would rather rely on the public repositories, and you will do your computational analysis, including meta-analysis or repurposing the technology. And actually, before I will kind of go to particular repurposing, I just want to kind of encourage you in terms of secondary analysis, as well as want to provide the scale. So when we thought about this secondary analysis, we thought, what is the scale? Like, do we know how many people in the world are doing secondary analysis versus primary, right? Like, what is the more popular as the technique to study the biology, right? Are people still in vast majority doing wet lab research, or really the secondary analysis, the dry research is taken over, but to what degree, right? So having that question, so what we did, we took the papers from the PubMed, several millions of them, and we parsed them. And we parsed them to the extent where we look for the specific data sets, SRA1 and GEO, and we were able to classify every paper as the secondary analysis or the primary analysis. And then we asked the question, like, how many papers are actually doing primary versus secondary analysis? So what you can see for SRA, still the majority of papers will do primary analysis, and some fraction, which I think is substantial fraction, more than 20, around 25%, will do secondary analysis. Actually, for GEO, there are slightly more papers doing secondary analysis than the primary analysis, which I think is remarkable, right? So it's remarkable that secondary analysis is really taken over in some sense, and then it's a new way to do research. So, and actually, we were also curious to look at that across the years. You know, when did that happen? When did it happen that secondary analysis became a more popular technique? So that did not happen, obviously, for SRA. The numbers are more or less stable across the year. So those plots are not cumulative. So the fraction between primary and secondary, that's per year, right? So that did not happen for SRA. And we can discuss why is that. I think there are reasons, we don't know for sure, but we kind of like think about few reasons. Why is that? But for GEO, you can see around 2016, the trend is reversed, right? There is more papers doing secondary analysis than the primary analysis. Okay, so what about the percentage of the data sets which were ever reused, right? So that's still low, I would say. For SRA, that's around 30% of the data sets were ever reused, and 70% were basically untouched, right, for the second time. And then for GEO, it's still high, 60% were not touched, right, as the second time. So why it's important? So that's important because to store those data sets, we're spending quite a lot of money, right? Like, and the whole purpose of GEO and SRA was to store the data for generation, right? Like any data generating in the world will be stored so people can look at that. So there's a lot of resources being allocated to that. But I think the purpose of those like repositories were just to store, and there is little discussion about like how to make it reusable. Right, and we can see that they're not quite reusable, right? Like people reusing that, but I think we can do better here. So I think the reason here is because there is no kind of like any initiatives or any thoughts about how to make them reusable. And for example, for SRA, we are storing as the raw data, right? We are storing as the FASTQ files on SRA, and I would argue that's probably not the best. It's a good format to store the data, but that's not the best format to make it reusable. Right. So for example, the BAM files will be much better. And the reason it's much better, for example, think about you're studying HLA low site, and all you just need the reads from that low site. So if you need to parse it across 10,000 samples, you would need to download the whole data, align the data, and like split the BAM file and get the HLA reads only from there. So, but that's so much redundant work, and everybody needs to do that. Right, so rather it will be better to store as the BAM files, but I imagine there are better formats like to do. So in terms of the superstar data sets, what we call, so here we looked at the number of times the data sets were reused. So you can see for GEO there are some data sets which are reused over 100 times. So what about those data sets? So actually one point of that project is to look for the properties which can predict the reusability. So in this case, the disease was quite informative. So I think all the superstar data sets in the GEO were coming from cancer. And I think the most reused one were coming from the cell line from the breast cancer. So as for SRA, it's not that clear. Somehow the most reused data set was the cotton. We don't know exactly the reason for that. Maybe just a single data set from cotton and everybody need that. We aren't clear yet. But in general, I think we observe some interesting kind of parameters which predicts the reusability, for example, sample size. So if the data sets you have generated has less than 10 samples, basically nobody cares about your data sets and people will probably not reuse it because it's too small. Also technology, right? The read length increases. So if your read length is 36, probably also it's less trustable or reusable. So there are multiple parameters we're exploring which can predict the reusability. Okay, so that's the kind of conclusion for this part, that reusability of omic data varies across different repositories, and in Geo it's much higher. So why is that? We think it has to do with the format and how easy to reuse the data. For example, for Geo, Geo is basically just gene expression. So essentially it's a matrix, right, where every row is the individual and every column is the gene expression, right? So it's not that big, so it's easy to operate. And people with limited background who are life science researchers, it's doable to download the data. And Geo provides a quite good online interface where basically you can go and click several buttons and generate all the individuals you need and generate that matrix and perhaps open that in Excel or any kind of other comfortable, comfortable software. So as this array, that's actually raw data, as I mentioned, that's FASTQ file. So to process the data, you would need some knowledge in bioinformatics to run the tools. Let's say you want isoform splicing or something like that. You would need to install the software, run it, download the data, have the computational capacity. So there's a lot of hassle, kind of in that regards. So as a result, many people just don't have skills. They don't have the time. So that's why I think that's one barrier. So I think interesting discussion should be launched how we make the format to be kind of like useful and then easy to use so that we observe more reusability. Okay, so that's about kind of a general introduction. So what I want to talk. Yeah.
I think you mentioned that many people do the same analyses, at least pre-processing, right?
Mm-hmm.
The backend is different, right? They do different analyses afterwards. It seems like if people do the same analyses on raw data, there's a lot of energy waste also. Right?
Yeah, absolutely. No, that's a waste of time, energy, on everything. Yeah, I mean, there is something happening. So for example, European DNA, like analogy of SRA in Europe, in UK, I think they store the BEM files, or at least some samples they store, but I think they only store if people who upload the data, they provide them. I think they will not routinely remap the reads. So I think one interesting project, I think we even talked to Mohammed about that, like taking the whole SRA and basically producing the BEMs and then storing them somewhere. And then I think the concern will be, what about the reference, like the version of the reference, right? Like if we get a new reference, what we do? Yeah, like is there some efforts to update them or should we remap it? You know, but I think it's interesting kind of discussion about the formats, right? And another format could be, like, for example, something on the k-mers, for like if you have metagenomics, right? So you have enormous kind of data, but let's say you care about only particular virus. So maybe you can pre-process the data to extend to provide the candidate reads for that virus. Basically, it will have all the reads from the virus, but some of them will be not actually from that virus, you know, like, but that's useful because you probably use the data like hundreds or thousands times. So I think that's, but I don't see that discussion happening in terms of like how we facilitate the reproducibility, in terms of at least the format, right? I think resources are there. I mean, at least if you have the money, Amazon is around, right? Or even on the cluster, right? The things are there, but the format is really something that can, like, exactly what you said, you know, can eliminate this redundancy of like data analysis, which is kind of ridiculous. Sounds like if everybody is remapping the same thing and nobody shares. And actually that happens to us. We were probing the whole SRA, and I think we ended up realigning all the reads, but somehow in the end, we didn't have place to store them, so we throw it away. But such a waste of resources, just because it was nowhere to upload them and share, you know, and we cannot store it forever on the cluster. So I think we ended up removing that. So that was, which was probably very ineffective.
Storage, how much storage you have in different formats,
right?
And then how much computation and data moments you have, right?
Exactly.
Yeah.
Cool. Okay, so today I want to talk more about computational immunology. So I think I would like to talk in particular about the adaptive immune system. So I think, as you know, there are two major systems: the adaptive one, innate immune system. So the way to study the innate immune system would be to look at the different cell types and look at the relative abundance and other properties. So that's something I will not talk about today, but I would rather focus on the adaptive immune system and talk about the challenges there and opportunities. So if you think about the adaptive immune repertoire, this is actually the repertoire encoded by several loci. So just to get the terminology, so we all on the same page. So we have T cell receptor, TCR, and we have Ig, immunoglobulin receptor, in other words, B cell receptors, right? So we have all those receptors distributed across several chromosomes. So, and then the mechanism is quite interesting. So if you zoom in a particular loci, so what you will observe a collection of the V genes, variable genes, D genes, diversity genes, and J gene, joining genes. They are distributed across that loci. So as you're trying to generate the receptor, you're allowed to recombine any V gene, D gene, and J gene, which we did here, right? We took V1, D3, and J2. But probably you have heard that the diversity of the T and B cell receptor repertoire is really huge. So it's like 10 into power 15, right? So that mechanism will not, given the small number of genes, right? That mechanism will not allow you to generate that huge of diversity. So what is also allowed to do, you're allowed to insert basically any nucleotide at the junction point, right? So those are called non-template insertions. And by doing so, you can actually produce astonishing diversity, and literally the immune system is capable of producing any sequence. Few definitions. So we will be talking about complementary determining regions, CDR1, CDR2, and CDR3. In particular, we will be talking about CDR3 region, because this is the most diverse, right? This is where the most diversity is captured. And we'll use CDR3 region as the proxy to define the clonotype. And the collection of all the clonotypes from a particular individual will refer as the repertoire. Okay, so those will be the definition. So what that means in terms of the reads, right? So if you're getting the reads entirely mapped to the V gene or the J gene, or actually the constant regions, they will be actually just fine aligned with any alignment tool you're using. And the reason for that, because V genes and J genes and the constant regions are part of your genome. However, obviously the recombinations are not. So the recombinations are part of the receptor of individual T cell. So this is what we're interested in. So we are really interested in the reads which are spanning kind of the recombination, right? Meaning the part of the read is coming from the V, some part from the D, and some part from the J, right? And those reads can be leveraged to reconstruct the CDR3 region, right? And essentially quantify your adaptive immune repertoire. So how we will do that? And actually before that, so the technologies you can study, it's actually, there is a proper technology, ARSeq. So that's a technology to study the repertoire. And then that's great of the technology. However, it's not scalable, right? The number of samples, I think, in the world will be several thousands, right? So not that much. And if you think about studying human immunome, right? So which the term probably you didn't hear so often, the human genome. And the reason for that, because it's way more complex and way more harder to assemble that, right? So when we did the human genome, that was straightforward in some sense. You just need to get a very nice reference of the particular individual and you're good to go, right? And the reason for that, just because we are more similar, right? We are so similar to each other, right? In terms of the genome. So it's enough to have one good reference, and then the whole field of bioinformatics or next generation sequencing analysis have started by realigning the reads from your genome to somebody else, right? Which is actually, by the way, a very strange biological problem, right? And when you're taking the DNA from one person and aligning that to another one. So it's a very technical one. And actually, for that reason, you will be never able to generate the real benchmarks there, right? Because it's completely artificial problem. So, but here it's way more complex because we are more different in terms of the human immunome than we are similar. So as a result, to really reconstruct the human immunome, in an ideal world, you will probe every single person on Earth, right? And get the human immunome. So that's not feasible yet. However, I would argue that RNA-seq technology is ill-equipped to do that due to the limited sample sizes. However, there's other technologies which are at the much larger scale, including RNA-seq. So we have around 70 or 80,000 samples sitting on SRA, right? So we can leverage those and try to reconstruct the human immunome. But those technologies need to be repurposed, right? RNA-seq was designed to study transcriptome rather than the human immunome. Okay, so what we did, we developed a tool called IMRAP. So that's alignment-free detection of the CDR3. So more details are in the paper published, I think, 2 years ago in Nature Communication. So the method is quite simple. So I will walk you briefly through the method. So the way we approach that is that we take the reads which are unmapped, right? Meaning the reads which were not mapped, right, to the genome, and we convert them to amino acids, to all 6 frames. So the reason we do that, because we actually have definition of CDR3 sequences in front of us. And if you look kind of long enough at the sequence, you observe some kind of fundamental properties that that's a sequence which always starts with cysteine and always ends with the phenylalanine. So if that's the case, so maybe we should look for those cysteine and look for the phenylalanine inside the sequence prior to everything we do. And I think that was one innovation of our method, because all other existing methods, you were using this information in the very end. So we thought, let's simplify this problem as much as we can, even maybe sacrificing some algorithmic beauty of the problem. And I think the previous methods, they were trying hard to assemble things using sophisticated and beautiful algorithmic structures and getting the set of the clonotypes. But in the end, they only consider something which starts with cysteine and phenylalanine. So we said, like, why waste time? Why don't we use these properties just the very front as early as we can? And this is what we did. So we take every single read, we scan it until we find the cysteine. And we do the same from the right to the left until we find the phenylalanine. Great. So this is our what we call candidate CDR3 sequence. So why it's not real CDR3 sequence, we never check the flanking region right here, right? We know nothing about them yet. So what we can do, we can take all the V genes and kind of align it one by one. And actually, we don't even need alignment. And the reason for that, because if you look again carefully at the FASTA file corresponding to these genes, you will observe that the 3 before last codon is always cysteine. It's remarkable. So what you can do, you can just put cysteine above cysteine, as simple as that, and check the match. There is no match here, no match here, match here. That's it. So the same we do for the J gene. We find the match, and that becomes our CDR3 sequence. We do some error correction by clustering, just to make sure like we don't assemble erroneous one, but that's pretty much the idea of the method. So another thing is that sometimes the reads are short, so as a result, the reads will not span the recombination, right? So the read will overlap V gene, but will never overlap the J gene because it's so short. So in that case, we record kind of partially CDR3 sequences, and we use suffix tree to match them, right? So there's techniques to overcome it, but that's roughly speaking the idea of the method. So doing that method, we were able to provide better results comparable to the existing methods, and we started, as typically we start in bioinformatics, with simulated data. We simulated extensive amount of the data across different read length, different coverage, and we see that we're doing quite good, actually very good for very short reads. And if you would like to summarize it as the F-score, the results are even more evident. Okay, so as we do simulated data, we always try to involve real data if possible. If there are real kind of data benchmarks, we would like to involve them. Okay, so here we took the BCR sequencing from 18 tumor biopsies, and those samples were sequenced by BCR-seq, which is the proper technology to study the adaptive immune repertoire, and we took RNA-seq. So, and what we looked for, we asked the question, how well we can estimate the relative abundances of the clonotypes. So what you can see on the x-axis, this is us. So we're using IMRAP by RNA-seq technology, and then on the y-axis, we are using the BCR-seq, kind of the gold standard or the ground truth. So you can see that we're doing quite well. We're able to differentiate highly abundant clonotypes and rare clonotypes, but in general, we're getting the good results. So another question we have asked, what is the fraction of the entire repertoire, of the entire collection of the clonotype we're able to capture? And we measured then that by the sum of all the frequencies of the clonotypes we're able to capture. So what you can see here, and then actually we divide that by the clonotype frequency, right? So if all the clonotypes would be above 0.06%, somewhere here, so IMRET will get them all, right? But as soon as the frequency of the clonotype becomes more rare, we get a harder time. So overall, we are capturing more than 50% of the entire repertoire. So why is that? It's obvious reason because RNA-seq lacks the depth, right? So because when you do RNA-seq, only the small fraction of the reads goes to the B cells. The majority obviously goes to the transcriptome. It's exactly opposite when you do BCR-seq. It's the targeted technologies where every single read will come from the clonotype. So we are not well positioned to capture the rare clonotype. But despite that, I think overall, RNA-seq is a suitable technology to study the human immunome. So as we got the method kind of validated, we thought of applying that to as large data as we can get our hands on. And that was the GPEC, Genome Tissue Expression Consortium. So that's a tremendous resource where more than 1,000 now individuals were probed post-mortem and across 50 body sites. So it's a remarkable resource, and we were part of the consortium. So what we did, we looked at the distribution of the IG clonotypes across different body sites. So obviously, you would expect the secondary lymphoid organs like the spleen and small intestine, terminum ileum, will harbor an increased diversity of the IG clonotype. And that was the case. We also use the brain and the nerve as the negative control. So in the brain, we expect to observe no clonotypes, right? It's an autoimmune organ. And that was the case. We observed a tiny fraction of the clonotypes in the nerve, and we believe that was the contamination from the blood as the tissue was prepared. But interestingly, we observed some kind of interesting group of tissues, and all of them have some properties, like they have mucosa sites being associated. So we think those IG clonotypes are kind of a mucosa-associated ones, and then they're quite diverse in that sense. So for example, this is what we never expected when we started the project, like minor salivary gland was harboring quite a lot of IG clonotypes, more than, for example, the terminum ileum. So that was interesting. So as we did that, we also kind of put that in the resource, which we share with the community. So, and that resource will help you to classify the clonotypes which are public versus private, right? So you might think that every single recombination and especially the insertion, right, should be personal, should be only occurring in a set of individuals. However, that's not the case. So some clonotypes are shared across the population. And then the reason they're shared might be that they might be shared across specific populations which were exposed to specific viruses in the history, right? So there is some heritability being involved in the adaptive immune system, but we cannot study that at the scale because we don't have the data, right? If you're using the proper technologies, and that's again coming back to this reasoning, if you're using the proper technologies like TCR sequencing or BCR sequencing, the number of samples will be not enough to see on the large scale, like what are the private clonotypes and what are the public ones. As here, so we provide that information across 3 million clonotypes we were able to assemble. Okay, so another kind of validation. So we know we have a pathology nodes for every single sample from the GTEx. We have a histology images and the pathology node. So we talk a particular disease called Hashimoto disease, autoimmune disease happening in thyroid. So what we have looked at the stage of the disease. So as the severity of the disease progresses, as more clonotypes we do observe in that tissue, in the thyroid. We also look at the sex differences across the samples, and we observe none, except the tissue you would expect to observe, and the tissue is breast. So where, as expected, again, a number of clonotypes in the males is significantly smaller than the females. With one exception, we were able to find an individual having the disease called ganymeoplastia. So it's basically when the male breast becomes like a female breast. So it's expected to harbor a substantial number of the clonotypes there. Okay, so that was kind of the method. So as the next step, we would like to really tackle the human immunome. So we'd like to take all the data we have in the world, which is 70,000 samples. I think now it's already 80,000. and repurpose RNA sequencing using IMRAP and get all the clonotypes we can and actually try to do human immunome across diverse populations. And I believe that the patterns of the clonotypes will be substantially different because different populations were exposed to different pandemics, to different viruses, and that is embedded in the system. But yet those studies to emerge due to lack of the data. So I think we are well positioned, having the largest sample size to answer those questions. And we have some preliminary results where not that we know the population, we also know the diseases, right? In the public databases, we also sometimes know the disease, and we can look at that. So for example, in this case, we just look at the regions, like something very simple. And we look at the enrichment of the regions across diseases, and we see some interesting patterns where, for example, for type 2 diabetes, we observe some distinct V genes to be present. So that's just the start of this analysis, and we're looking kind of to scale that and look at different diseases and different metrics. Okay, so as we did that, we also thought about the diversity in monogenomics, and there is quite little known about that. I would argue in kind of in normal genomics, we know more about that. So we know about polygenic risk scores, how those are not well adjusted across the population. If you're computing your polygenic risk scores on Europeans, you obviously cannot apply them to other ancestry groups. We also know in genomics what is the fraction of the data which was generated from European population versus others. So I think it's 80% or something. So we know that. So nothing like that was known in immunogenomics. And we thought that's a great place for us to contribute, so which we did. So we published recently 2 papers, one in HMF and another one is cell immunity, when in one we just kind of provide the framework on the like the paper on the left, so about like why diversity is important, what can be done. And here we actually do the analysis. We do the analysis, what is the distribution of the data across the population. So, and then I will start with that one where, as you can see, that the vast majority of samples are coming from individuals of European ancestry. However, that's based, if you count based on the sample. However, if you look based on the study, there are actually a little bit less fractionally number of studies. I mean, there's still majority of European studies, but for example, Asian-based studies are higher comparable if you compare to the number of participants. So why is that? And actually the reason is because European-based studies just have a larger sample size. So we have shown that. Then we have look, and the way we did it, so we look through the papers and we look to see if people do share ancestry, right? Self-reported ancestry. And this is the distribution. So you can see that only less than 20% of the papers share the ancestry in the publications. So another 20%, those are good people willing to share it. If you ask, right, you email the author and say, hey, can you please share that? And they do. But the vast majority, it's either they don't want to share for certain reasons, which I will discuss, or just not available. They're not replying the emails. So it's actually interesting question about sharing the metadata, right? So we are quite agreed what was the framework to share the raw data, but I think we're quite far away in terms of the metadata. So this is what we published recently, how we improve that, right? Like how we improve the completeness of the public data, which is not, that's far away from being good. I think overall, only 60% of the metadata is shared and 40 is just lost. So, but that's crucial because, for example, your study, let's say you do ancestry-based analysis. So if you don't have ancestry of those samples, for that particular analysis, despite having the raw data will be useless, right? So I argue that we need a better kind of the protocol or conventions to share the metadata. And then actually, interestingly, some people indicated why they don't share. I think there are a few reasons for that. One is cultural. People don't see the value, right? They basically say, why would I? What's the point? And I think it's understandable because when you work with your data, let's say everybody is Europeans, you don't see the value to saying that everybody is Europeans because in your setting, in your world, you don't account for ancestry because you have homogeneous population. However, as you share, that's not longer the case. So you need to know that ancestry of those individuals. Sometimes IRB, internal revision boards, if you didn't say that you will collect ancestry and share that, you cannot do that. So when you design the study, should be better mechanism to kind of to ensure good design. Otherwise, you're just locked. Maybe you already want to share that, but you can't. So we argue that probably we need to change that. Maybe instead of the current model, when you design the study, you say which phenotypes you would like to collect, right? That's the case. So we argue that perhaps we need to reverse it. So you need to argue which phenotypes you don't want to collect and what is the reason for that. For example, say, I don't want to collect ancestry. You should say why, right? Because this way we can define the minimum set of the phenotypes to be shared and everything will be harmonized in terms of the metadata. But that's far away from being the case. Okay, so a few more things before we'll wrap up. So another problem, the current immunogenomic databases are disproportionately incomplete. So why is that? So the reason for that, because the immunogenomic database were built historically on Europeans, in vast majority, on Europeans population. And then as a result, they serve not well other population. And that's more severe, I would argue, than the human genomics, where let's say if you build a reference on one individual, on one ancestry group, on one population, and you map the risk from another one, I think that will be still okay, right? You will get more mismatches, maybe things will be harder, but overall the results will be feasible. As here, if you're lacking, there's more differences across populations. So if you're lacking the particular V gene in the database, and that V gene is present in the population, you will never be able to discover that or observe that or quantify it. So there is a pressure need to do that. So, and the question is, can computational immunology help? Like, can we leverage sequencing data to infer novel alleles? And what we're suggesting to apply bioinformatics method across diverse public data to infer novel gene alleles, like the alleles of the V and J genes. That's yet to be happening. So at the moment, there are several tools which are there. However, the relative performance of those tools is unknown, or at least the way tools were benchmarked inside the publication are not comprehensive, at least to our opinion. So we would like to perform the benchmarking studies of how well the TCRC can be used to inform novel TCR allele, genes and alleles. And for that, so we previously formulated the principles of the way to do benchmarking of omics data. And I think when there is a challenge, there is always opportunity. So we consider that's opportunity for us to contribute to the field. So we'd like to benchmark using the comprehensive gold standard. So how would you design the gold standard? I think one way to do it, just to do simulations, right? That's usually how you start. However, I would argue that simulations are never can capture the reality of biology. It always will be oversimplification, which is fine as the starting point, because you actually have quite a flexibility to generate all the scenarios you would like, right? So you don't have any kind of restrictions in terms of scalability. You can do as much as you want. So what we're suggesting, we're basically suggesting to take the full database, simulate the reads from the full database, then artificially remove, like make it incomplete, and ask the question, can you reconstruct the ones you removed? Right, very easy. You can vary the degree to which database is complete, right? You can ask the question, if the database was initially 20% complete, can you restore 80%? Probably not. But if 50%, can you restore the remaining 50%? So you can really kind of do that. However, again, that's simulation. So what we're thinking about to extend it and try to get the real data, which we already started. So we recruited 10 patients in Los Angeles. So luckily, we live in the heterogeneous area, so we don't have any issues recruiting diverse population groups, including Asians, Latino, and others, so which we did, and we would like to use real data. So that's done. So then we have run the AR-seq data, in particular TCR sequencing data, and we are in the process of running the tools. And I think the most challenging, that's kind of straightforward. The challenging part, how would you validate them, right? How would you know that those alleles which you assemble are true? So what is often happening is like you can do PCR, or you can do Sanger sequencing. But the issue is like for how many, right? Like let's say you got the thousands alleles. How many would select do PCR? And unfortunately, the very common approach is actually to randomly select, set a number, and send it to PCR. And the message you often see in the kind of those papers, you see, okay, we selected 10 alleles or 20 alleles, And we got lucky, all of them are true, right? So we would argue that's not a comprehensive approach. You would need to validate every single one. Actually, beyond, you should validate not only the ones you observe, but the ones you don't, because otherwise you cannot do sensitivity. And I mean, you can do precision, right? If you validate every single one you have assembled, then you can do precision. But how about sensitivity? You can, because it can be unknown ones. So how we actually validate the unknown ones, right? That's a big challenge. So the way we propose, yeah, so like here, right? So false negative. How about false negatives, right? Like that's a big challenge. So what we are proposing, a kind of approach which we think will be able to do false negatives. So this is the way we'll do it. So think about like we got all the alleles, right? And actually those in orange are the alleles captured by RNA-seq samples. So what do I mean by captured? They're just covered by a single read. So I would argue that if the allele is not covered by a single read, you will never be assembled, right? So we can ignore all the alleles which are theoretically exist, but not capturing your sample. So, right, so we'll only focus on all the alleles covered with one read, which is feasible. Basically, you beyond those methods you kind of run, you also take some simple mapping tool and you map all the reads against the loosely, you map very loosely against the database. So every single potential match you will report as the candidate allele. Then I would argue you have a complete phenotype. I'm sorry, the complete set of the allele, at least in the regards of kind of sequencing sample. And then we'll treat that as the complete gold standard, and then we'll do false negatives. So actually, we did a similar approach for structural variance benchmarking, where actually the data was not done by us, but our collaborator, they took chromosome 19, they identified every possible deletion by eye, and they went to the lab and validated every single one. I think they took it like a few years, but that's really complete gold standard. Okay, so just like to be on time, and so I think the barriers in the data-driven immunology are the following. So lack of computational capacity, inability to perform bioinformatics analysis, control access to the data set, and incomplete data set, incomplete metadata. So some of them can be overcome. For example, if you think about inability to perform bioinformatics analysis, let's say for the people from immunology, from the life science background, I would argue that can be overcome with the appropriate training, which we formulated before. So that can be done, right? So that's people, I mean, the researchers can be educated and empowered by bioinformatics skills. However, something like incomplete metadata is harder. If the data is incomplete, I mean, there is no really a way to infer it. Some of them, actually, some of the metadata can be inferred from the sequencing data. And actually, we are working on the method to infer the ancestry from TCR-seq data. Right? It's unclear if we can do that, but at least we try. And definitely, I would argue ancestry can be inferred from RNA-seq data. So there is an interesting component that you can derive some phenotypes from the raw data and maybe correct the errors or misreporting in the like biological sex. Obviously, you can, right? So what we observe, so we took, I think, 3,000 samples, maybe, I think more, 10,000 samples, like 30 studies across 5 diseases, and we look at the completeness of the metadata. So what we mean by completeness, we look at the original publication, we read it manually, and we extract every single phenotype, as well as we take the public repository and we look at that file. And then we take the union. And we say, here, for example, ethnicity is widely unreported, right? So like around 60% or even more, 70% is not reported. The same for the mortality and clinical severity, things like that. So that's a big problem, because unless you contact the authors and they are nice to tell you the metadata, there is no way you can restore it. Another story is that if the data is shared only in the original publication, but not shared in the public repository, potentially you can run, let's say, natural language processing or something. You can kind of restore the phenotypes from here. It's questioned with what accuracy. And kind of complement the public repository. So here is actually interesting. So we contrast how people share the phenotypes in the original publication versus the public repository. So you can see that on average, there are better sharing in the publication, in the text of the publication. There is some kind of gap, lack, when we do public repository. But there are some exceptions, you know, like sometimes people do, I mean, more common people do better job here, right? But there is some exceptions. For example, people don't share very little in the original publication, but suddenly they share very nicely in the public. So there is all sorts of like trajectories here. Okay, so just like to wrap it up. So I think I would argue that the open data and bioinformatics methods are powerful solutions to immunological research. And actually what is interesting, they're suitable for the countries with the limited resources, right? Like there is no need. I think that the whole concept of the dry lab is extremely appealing, or I hope will be appealing soon for the countries with limited resources, because you don't need to build the infrastructure, right? All you need is the knowledge, the training. And we have argued about that few years ago, that it's a suitable model for the countries and actually universities with limited resources. And I think it's like another piece we are working on. So that's a preliminary draft there. So kind of what we say, the path to independence. So that the dry lab are really taking the independence, which they were traditionally lacking, right? And I think that relates to what Pavel Pevsner was saying, like, is the bioinformatics like a servant or the queen of biology, right? So like that. Okay, so that works of many. So many students, excellent students in my lab at UC, and we're always hiring. So if you're interested to do postdoc in sunny California, so please contact me. Okay, thank you so much.
I have a question.
Yeah.
So reusing the data is always great, right? Data costs for sequencing and generating the data, but it has its own challenges as well, right? So you need to develop new tools that maybe not needed when you do the sequencing from scratch. For example, if there's a new sequencing technology coming up, and they provide much better throughput or higher accuracy and so on, if you want to use old data, you may still need some correction tool or those tools that you publish to compensate for this accuracy loss. Right? The same thing with maybe to do a remapping again or do the analysis again. You might find a better tool to do the things much more efficient than just repeating the step from scratch. So, which one is still better?
Yeah.
To develop these tools, wait until we got a bunch of all these tools for different purposes, or just pay for the cost of the sequence.
Right. Yeah, yeah. No, that's a great point. So yeah, I think it's like the main limitation of the kind of dry lab. You assume that the data is available, right? Like if you you're limited by the data, right? If there is no data from this disease, you cannot study that, right? And exactly as you said, like if there is emerging technology appearing, you have two kind of solutions: either like get some money and then like sequence that, or wait until there is enough data being accumulated. But that's always like kind of, you're always constrained by data availability, right? So that's always the challenge. But I think that will go away as we scale. And sounds really scaling, like sequencing is really like taking the pace, right? So we'll be seeing more and more data. So that will, as we accumulate even more data, that will kind of go away in some sense, Yeah, but we will always lack, as you say. If there is a new technology, there is a lag between the technology appears and we accumulate enough data. Probably like nanopore, right? We don't have publicly enough data sets to do anything like population scale yet, right? But that will probably happen at some point. Yeah. Yeah.
We have a question.
Well, I have actually several questions, and mostly like in the personal population. So when you show the trend regarding the usage of the primary and the, let's say, the secondary usage of the sequencing data, right? And it seems like there's a trend that the secondary usage is sort of increasing. So it was like sort of counterintuitive to me because like the cost of the sequencing using sequencing technologies is also decreasing. So I would assume that like more people are basically sequencing their data and let's say uploading them to the cloud centers, and then like they are basically primary data. So what I would expect like so we will have like more and more primary data.
Right, which is true. I think is like, so yeah, so basically that's a great point. So I mean, I need to find a plot where I mean, I don't have it right now, but like absolute numbers, right? Like this will be like, yeah, but that's true. I think that's true what you're saying. So basically, exactly, like we're generating more data. But I think proportionally, secondary analysis is even more than. So like so it just like, it's a matter of like, it's so accessible or it's so easy to do, right? And actually that's another challenge, like how we avoid like kind of sloppy science in some sense, like these trade mills where like you just do secondary analysis, you produce the paper and so on. And that's a big concern then when people say the sentiments against secondary analysis, because many people who generate the data, they don't think the people from the dry lab have enough understanding of the disease and so on, so they to be able to perform, which sometimes perhaps is true and sometimes not. And that's why there is this sentiment, which is like data parasites and things like that. So that's, I think, just because it's emerging, and as you can see, it's emerging on such a fast pace that perhaps makes some traditional wet lab uncomfortable. But I think you're right. Like there is more data being generated, but the secondary analysis space is even higher. Yeah.
This is sort of also related to my second question. So, like, I guess anyone can go and let's say to an ENA and submit their, let's say, raw data or test data. So there is no basically rule or restriction, I guess, for me to not submit
right.
if I have my data. So do you think like we should have like certain, I don't know, restrictions or quality control or like any like format checkings when submitting such data? So how do we know that basically this is like data that is really generated for that like certain purpose, like in that certain?
Yeah, no, I think that's a great question. So yeah, I think so, and I think it's an open question how we do that. Like the most simple will be, for example, to check for the species, right? Like which is trivial, right? Like that you don't upload the wrong species. So that's trivial. But for example, can you really prevent some fake data, right? And I think that's an interesting question. That's an interesting research question. Like, can you basically can you have a model? Let's say you go and train that on the real data, right? And then you have certain expectations of the data, right? And that model can check if the data is real. And then on the opposite, can you kind of trick it, right? Can you generate very sophisticated data, so that the model will say it's a real one? But I think that's a big problem, because I would imagine you can, like no matter how kind of sophisticated you are to train your data, you can always take some another real data and change it a little bit, that it will look like. And then, so that's a big question, right? Like, and that's in general big question about public data. Like in addition to incomplete metadata. So I think there are 5%, maybe less, 3% of the data sets where biological sex is mislabeled, which is a disaster because it's such a simple phenotype. Like if you cannot report the biological sex, what about more complex phenotypes? And luckily, you can check that from the genomic data, obviously, from the RNA-seq data, right? If you just look at the chromosome, XY chromosome, and you got it. And I think it's very accurate. So I think that needed, we should, I think it's like a lot of QC quality controls needs to be done for the public data. You cannot just like trust it. And then I think the good part, because you typically work with the large sample sizes. So as a result, if you don't feel comfortable or you think you have some suspicious about some data set, it's always better to throw it away, and then focus on the data which you trust and it seems like high quality. So in that sense, yeah, that should be always. I don't know where should that happen. Should it happen like as you are saying on Serghei or ENA? They should do it, or is it? But at least it's that is not happening there. That is responsibility of the people who are performing secondary analysis to kind of ensure the quality control as much as you can. Like not trust anything at all. Like even the metadata. Like if you can verify that, please do. If you have some distribution of the genes or whatever which is abnormal, like it's better to throw away that particular cohort. Probably you will have still enough. But that's a good point.
Similarly, NCB has actually given some sort of similar scores to like certain species, I guess.
So again?
So when you basically submit your data or like searching for data, you can still like find some sort of information regarding like how similar is that your sequencing data to a certain species, let's say. But
yeah.
This is like a basics, like
right. But there are more than that. There are like distribution of the gene expression, distribution of the mutations, you know, we can take that to the new level in that sense, you know, like I think there are some kind of profiles of the gene expression, profiles of the mutations, expected one. So if you're the one you're submitting is not like that, maybe it's still true, but then perhaps it needed some manual check or something, right? But at least if it's within that distribution, that perhaps some indication the data is correct, right?
Do you have a more detailed breakdown of what kind of formats people usually use for the secondary analyses? Because if you come up with, let's say, a distribution of the most popular formats, that may be.
Yeah, but I think if you think about sequencing data, that will be always, I mean, it will be kind of FASTQ files, like what is this, the format on SRA? I forgot, like there is their own format, but essentially raw data. There's no much variability in the formats.
But they do some analysis on the raw data, right?
Cool.
And then they create some intermediate formats for themselves, perhaps.
Like this, people who do secondary analysis.
Secondary.
Oh, that's a great point. Oh, I see. Oh, yeah, that's a great point. So basically, you are saying is like if we look at the people who perform secondary analysis, what they do,
exactly.
Like what pre-processing they do, which could be done there, and then if you identify enough people of doing the same thing, you can argue that should be like
That should be your new format.
Right, new format, just like kind of asking the crowd in that sense. Yeah, that's a great point. Yeah, maybe you should do. Yeah, exactly. Just like looking what people actually need retrospectively. And then kind of saying, okay, that's like what people need like in 80% of the time. So perhaps that should be adopted.
Exactly.
Yeah. That's a great point. Yeah, no, I think so. Yeah. No, we have the data. We should do it. I mean, we have all those papers sitting. So we just need human power to do it.
Okay. This was great. I mean, please continue.
Yeah. Oh, yeah. Okay, cool. Yeah, thank you.
Actually, following that discussion, I would assume that, let's say, the PAM format is, let's say, the most common format that people are generating in the secondary analysis. So, like, maybe like being more conservative and saying that you must also submit your PAM file.
Yeah.
You're submitting FASTQ, and also saying that you must use that certain mapper, certain version, certain parameter.
I mean, the mapper is harder, I think, right? It's more ambiguous.
So would that also provide some advantages regarding, let's say, like concatenating all that data, like the PAM file submitted to the data center, such that you could actually compress in a meaningful way, like rather than basically storing all these PAM files like separately. Like,
but what
sort of similar to like performing marked up duplicates in PAM files. So you have like all these different files to the same species, and then but you know that like I don't know, millions of reads aligning to the same function, but you could maybe do some clever.
Yeah, I think it's like CRAM is doing partially that, right? Probably, right? Like they are smaller size than the BAMs. So that's something they do. So, but that's interesting question or direction of the research, how you compress the data, right? So, but I think it's like there are two questions. Like one is how to compress it, right? Which is like, yeah, it's interesting kind of area of research. I know little about that, but yeah, I agree. Like, and somehow people from SRA, like, I mean, I talked to them a few times, and my feeling they're really overwhelmed by what they do. And then it's also risky from their side, you know, like what about you're losing something? You know, like I think they're paranoid about that. So for them, it's better to spend like 100 times more storage or something, and then keep the things are because they are working. And then since they're overwhelmed, I don't think they have time to do research to ensure the new format will be not result in losing any data, right? Because for them, they don't care about reusability. And then this is what we hope to introduce here. We want to introduce the metric of reusability, right? So now we're relying, when people do good job, like doing the paper, you got citations, right? That's primarily the model, right? Like you get acknowledged. How about the data? How about, and people don't necessarily cite, unfortunately, there is no this good culture. If you're reusing somebody else's data, I mean, you would put the ID, you know, like, but you would not, let's say, cite. So we would like to introduce what is called like something like reusability index, and actually cheer those people, have some website, and have all those guys, the data. And I think it comes to what's his, Eric Lander, who's like now the scientific advisor of the US president. So I think he said like it's not about what you do with the data, it's about what the community will do with the data. Look, that's kind of motto. That like really cheering the people who generated the data 10 years ago, now it resulted in 1,000 publications. I think that's remarkable, you know, like the effort you took to generate the data resulted like kind of so broad, you know, like And then so that's one part. But another part, when you talk about like, that's a great point about read alignment, right? Can we enforce everybody to use that? That's a big problem, because I don't think you can, and I think that will be not right to do, because, for example, for the read alignment, the best benchmarking you can do will be always simulations. There is no possible way ever to do molecular based. And the reason for that, again, this is an artificial problem. It's not a biology. Taking my DNA and mapping to yours, there is no biology there. It's a completely artificial, it's a convenient thing to do. And then it was remarkable kind of discovery to do that, but it's not biology. So you can never know the true position of the reads through molecular techniques, like impossible. So all you do is rely on simulations. So now the question, do you really know what is the best read alignment, right? So, and then will it be okay to rely on simulation data to enforce everybody? So probably not. But then there is a variety of the aligners people are using. And then it's a problem when you do secondary analysis, because in my opinion, it's okay if bioinformatics tools are biased, but as soon as they're biased in the same way, right? Like if you process the whole data with one alignment tool, sure, there is a bias, but more or less is randomly distributed across all the samples, that's okay. But it's much worse than you have a variety of aligners, so the bias is like all over the place. So the question, but who need to process it? You know, like, so perhaps some third party, you know, like will take all the data, will take alignment they think is the best, will use the best settings they can, and reprocess the whole data and store it for somebody willing to use that, right? But I don't think we can rely on people doing that. So it should be like centralized effort to do that.
I like it actually based on the bias we have from the remapping tool. How do we know which one to use, which pipeline?
Right.
You have combination of both tools, not about remapping only. You have also downstream analysis.
Yeah. No.
The data is so challenging.
Yeah. Yeah. To me, it's like benchmarking is partially the answer. You know, especially for the domains where you can generate the benchmarking, right? So you more or less can know as best as you can do. You know, like the limitations of the tools, of the biases, right? Like something like kind of deep, comprehensive benchmarking. So that's the best kind of like the best we can do. But unfortunately, for some domains as here, you know, like you can't, right? So like the question then, what tools do you use? Yeah.
So yeah, let's thank Serghei once more again.
Thank you.
Hopefully within 30 minutes to 1 hour. I think we'll have a group meeting this soon. If we couldn't make it before 2, we move to another building. I think we have it in the calendar. So see you either here or in the next room in 1 hour. So we'll have lunch. Please don't miss the applause. Yeah, thank you so much.
Yeah, thank you.
Second